Hello everybody, my name is James Nicola and I am doing a video prompt on CRISPR for my Biology 170 online class for UW Superior. The prompt is asking how discovering CRISPR, the process, is as groundbreaking and innovative as dynamite by Alfred Nobel. Nobel Prize. So, and it also asks what are some potentially good and bad scenarios with it, and what is it. So CRISPR is a very long acronym, but more or less what it's saying, CRISPR-Cas9 is a way that a protein, the Cas9 protein, and the guide RNA go into a specific part of a gene, of a section of DNA, and make either a specific, well, it makes specific breaks right in the lines. If you hit bing, 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 cut here, it will either A, stop the DNA chain at that point, and then the body will repair it, or B, if you make a cut here, you cut that whole piece up. And if you wanted to, you could add stuff in. So doing this kind of genetic modification, compared to the previous version, is a lot more successful. 80%. 80% is pretty darn good. Now, what does this have to do with anything? Like, why is this applicable? So, you're manipulating genetics, the blueprint of, well, everything. If it has cells, it has DNA. Or RNA if you're, single cell organisms and whatnot. But, for example, animals, plants, and humans, we all have DNA. And being able to do these edits in DNA is huge. Well, the bacteria use it too as a self-defense mechanism. You could do multiple great things. I mean, the possibilities are endless. You could cure diseases. You could bioengineer animals to maybe produce more milk for cows, or more resistance to their environment. And same thing with plants. You can make them where you don't need insecticide or herbicide anymore. It's kind of built in. Or you find weeds and you cut out the parts that make it resistant, so any weed killer you put on it will kill it. Now, on the flip side, it's very controversial. I mean, you literally can write DNA however you want based on this. You could, do some kind of like a genetic terrorism, introduce and modify viruses or human genomes to more susceptible things, and unfortunately it only has an 80% success rate. In doing so, for example, in the video here, it could fail. Thankfully, after a lot of tests, trial and error in the breeding that we had, doing pigs and then trying to make human organs out of them, it had over an 80% success rate, which is great. And then after that point, you add in stem cells to get rid of that opening that you created by getting rid of the pig DNA, and adding human stem cells to create human organs. That's awesome. But, then there's the ethical part of kind of making it more human by adding human stuff. What if the stem cells go to the brain, make it think more human? It's kind of in the hand of the editor, whether they want to do good or bad with it, but the possibilities are endless. That's all I got. Thank you.